Hello, my name is Tessa Fuhrer. Welcome to the audio slide presentation of our recent paper, titled Impaired Expression of GABA Transporters in the Human Alzheimer's Disease Hippocampus, Subiculum, Entorhinal Cortex, and Superior Temporal Gyrus. I will spend the next few minutes providing a brief overview of our findings. Gamma-aminobutyric acid, also known as GABA, plays an important role in the regulation of neuronal excitability and acts as the main inhibitory neurotransmitter in the brain. The removal of released GABA from the synapse is dependent on reuptake by sodium and chloride-dependent high-affinity transporter proteins located on neurons and astrocytes. This study focused on the GABA-specific plasma membrane transporters GAT1, GAT3, and BGT1, all belonging to the SLC6 family of genes that have varied distribution in the regions and cell types of the brain. The aim of this project is to investigate the region and layer-specific density of GAT1, GAT3, and BGT1 in the hippocampus and superior temporal gyrus of control and Alzheimer's disease brains. We used a standard free-floating immunohistochemistry protocol on 6 control and 6 Alzheimer's disease human cases. 2 sections for each case from the hippocampus, entorhinal cortex, and superior temporal gyrus were stained for GAT1, GAT3, BGT1, and co-labeled with NUN, GAD, or GFAP. Images were taken using a confocal laser scanning microscope, and image J was used to measure the layer-specific transporter density within the hippocampus, entorhinal cortex, and superior temporal gyrus. In Alzheimer's disease, we saw a significant increase in BGT1 expression in the Oriens layer of the CA2 and CA3, in all 3 layers of the dentate gyrus, and in the superior temporal gyrus. BGT1 immunoreactivity was highly localized surrounding the pyramidal neuronal cell bodies in the hippocampal CA1, CA2, and CA3 regions, and the granular cell bodies within the dentate gyrus. In the dentate gyrus, we noted that the strong increase in BGT1 expression in Alzheimer's disease cases was due to an upregulation of BGT1 along astrocytic processes, shown in green, compared to control cases. The entorhinal cortex and superior temporal gyrus showed decreased expression of the GAT1 transporter in Alzheimer's disease, and as shown in the representative image, GAT1 displayed a relatively diffuse pattern of staining. The subiculum, entorhinal cortex, and stratum pyramidal of the CA1 and CA3 regions revealed decreased expression of GAT3 in Alzheimer's disease. GAT3 expression was more localized near the neuronal cell bodies. We did not observe a significant reduction in GAD immunoreactivity between Alzheimer's disease and control cases in the entorhinal cortex and the superior temporal gyrus. This may suggest that reduced GAT1 levels in these regions are not due to the loss of GABAergic cells and terminals. We also examined the relationship between GAT levels and beta-amyloid, tau load, and neuronal loss as possible contributing factors to changed GAT expression in the Alzheimer's disease brain. We found no significant correlations between any of these pathological changes and GAT levels. To summarize, we showed alterations in expression of the GABA transporters in the Alzheimer's disease brain, and that the expression of the GABA transporters show brain region, cell layer, and cell type specific alterations in Alzheimer's disease. Interestingly, where we saw the most significant increase in BGT1 density in the superior temporal gyrus is also where the most significant decrease of GAT1 density occurred. This could be depicting a compensatory response of the GABAergic system to regulate levels of GABA in the synaptic cleft, and perhaps the alteration in one transporter expression might be driving the change in another. However, the mechanisms behind these changes and the associated implications are still under investigation. As there was only a single previous study looking at GAT1 expression in cortical human tissue, it was very important to investigate the expression levels of these transporters in the brain areas that are most affected in the disease to advance our understanding of the underlying pathology of Alzheimer's disease.